Fighting Blindness Canada's Viewpoint is a virtual education series that brings you the latest in vision research presented by health experts from across Canada. The webinar you're about to watch is a recording. To learn more about the research we fund and upcoming webinars and events, please visit our website at fightingblindness.ca. This Viewpoint webinar is proudly presented by Bayer and supported by Allergan, Janssen and Novartis. We'd also like to thank Accessible Media Inc., our national accessibility partner. Everyone here should download the AMI app, which is a fabulous resource. And you can watch excellent programming, including previous Fighting Blindness Canada events. Thank you for watching. We hope you enjoy this webinar and share your thoughts with us in the comment section below. All right, so now we're going to move on to our Clinician-Scientist Emerging Leaders. So this past June, we announced the 3 recipients of the 2021 Clinician-Scientist Emerging Leader Award. This award was established in 2017, and it's given to early career clinician scientists who are pursuing a research career to complement their clinical practice in the field of vision research. So we want to thank all of our donors that have supported these awards, Andrew and Valerie Pringle, Dr. Peter Curtis, Bayer, and the Bank of Montreal for just being so generous and making these awards possible. Today, we're just really excited because we have all 3 of these visionary scientists here to tell us more about their particular projects. So I'm going to turn it over now to Dr. Larissa Moniz, who is our Director of Research and Mission Programs, and she's going to speak with each of our scientists a little bit, learn a bit about their projects, and then later on I'm going to come back and we're going to answer any questions that you have about their projects. So do remember, you can put them in that Q&A box or send me an email at education@fightingblindness.ca. So Larissa, I'll turn it over to you and our scientists.
Thank you, Morgan. It was actually really nice hearing about all those events, especially the 50/50 raffle. As a staff member, I'm not allowed to buy tickets, but I hope everybody else takes advantage of it. It sounds like some really great prizes there. I'm going to start off the session talking to Dr. Delphine Gobert, and then we'll talk to Dr. Tina Felfeli and Dr. Stephan Ong-Tone, who is currently, I think, doing something clinical and important and will be joining us shortly. But I'm really, really excited to actually have a chance to talk to all 3 of them today, learn a little bit about their research, a little bit about why they have chosen to become clinician scientists. I think it's going to be a really exciting conversation. So Dr. Gobert, I think we can just start off pretty simply. If you maybe just tell us a little bit more about sort of your career path. I know that you did a PhD and you did some postdoctoral work in neurobiology before you decided to go back to school and do med school and do ophthalmology. So sort of how did that career path happen for you?
Hi, everyone. Nice to meet everyone. I guess it all happened kind of naturally. I was doing research, and as I was doing my postdoctoral training, I was working on the visual system development. And I guess this is where I've first become interested in ophthalmology when I decided to go back to school and actually go to med school. And while I was pursuing med school, I found that ophthalmology was even more and more interesting, which is why I decided to apply to ophthalmology. And therefore now I'm a second-year resident at Laval University in Quebec.
And is it hard to sort of balance the demands of doing your residency with trying to do some clinical research as well?
Yeah, for sure. It's a challenge. Like, as you may know, residency is quite busy. I'm sure my colleagues would agree on that. So it's always hard to find time to do research, especially when it involves like spending time in the lab and working on something that is like not of clinical use on your day-to-day practice. But at the same time, this is what keeps it interesting to me.
That's great. So maybe you could tell us a little bit about your project. So I know your project focuses on a disease called diabetic retinopathy. So just to start out with, could you just explain a little bit about what this condition is and how it affects the eye?
Yes, sure. So diabetic retinopathy is a condition that can develop in people with long-standing and poorly controlled diabetes. So in diabetes, as you may know, circulating blood sugar or glucose levels are elevated. And as a result, a sugar byproduct can accumulate on the circulating red blood cells and on the inner part of the blood vessels throughout the body. And the smaller vessels, which we call capillaries, are particularly vulnerable because accumulation of glucose can more easily occlude and damage those very tiny blood vessels. This is why the eyes, which contain many small capillaries, are preferentially affected in diabetes. Accumulating glucose can also cause the blood vessels to swell and become leaky. So in diabetic retinopathy, there are actually two main forms, a non-proliferative form and a proliferative form. So in the first non-proliferative stage, accumulation of circulating sugar byproducts can damage the capillaries in the eye, leading to the formation of balloon-like structures in the vessel, which are called microaneurysm, as well as small areas of bleeding in the retina, which we call dot hemorrhages, as very small areas of bleeding. Now, if the capillaries become occluded, some portion of the retina may not receive appropriate blood flow, which can cause ischemia to the retina or lack of oxygen. And since the vessels can become leaky, fluid can also accumulate outside of the blood vessel and cause edema at the level of the macula, which is the part of the retina that is responsible for central vision. And as the disease progresses, there will be more and more areas of ischemic retina, which will drive the secretion of new molecular signal to grow new blood vessels in the hope of restoring adequate blood flow to the retina. This is the proliferative stage in which tiny new blood vessels are formed on the retina and into the vitreous, which is the gel-like structure that is filling the eye. But these new vessels are more fragile than normal blood vessels, and they can easily rupture and bleed. So in this phase, there can also be proliferation of fibrovascular tissue, which can cause traction on the retina and lead to retinal detachment.
Okay, so that's actually a great explanation. So if I have this right, so it's actually the glucose itself, which is damaging the vessels. It causes a lack of oxygen in some parts. And so then the eye is actually trying to sort of compensate for that by creating new vessels to bring in more oxygen. Is that correct?
Exactly. That's why the eye is trying to grow these new aberrant blood vessels, which are very fragile.
It's funny how the body's sort of trying to help out a little bit there, but it ends up harming as well at the same time.
That's right.
Yeah. So I think you already sort of talked about the blood vessels and how they grow, but if somebody has diabetic retinopathy, what sort of symptoms would they see when all of this is happening in their eye? And could you tell us a little bit also about maybe who's most at risk for developing the more proliferative stage of the diabetic retinopathy?
So diabetic retinopathy can produce a wide range of symptoms, actually. So as I mentioned previously, there are two forms of diabetic retinopathy, so the non-proliferative and the proliferative form. So in the non-proliferative phase, patients actually often do not have any symptoms unless there is associated macular edema, which is, as I mentioned previously, when fluid accumulates in the central portion of the retina. So if there is macular edema, a patient can report having decreased or blurry vision. In fact, diabetic macular edema is the main cause of decreased vision in patients with diabetic retinopathy. But since the early stages of diabetic retinopathy don't always cause symptoms or can cause only very mild symptoms, it is especially important for people with diabetes to undergo regular complete eye exam to make sure that actually there are no signs of diabetic retinopathy in their eyes. And now, as the disease progresses into more advanced stages and into the proliferative form, patient can develop those tiny aberrant blood vessels that we talked about, and they can bleed more easily, which can cause vitreous hemorrhage or bleeding inside the eye. And so when this happens, a patient can report a significant and rapid decrease in vision. Sometimes it could be just floaters, like little things floating in their eye. They can also talk about the shifting dark clouds or just like a sudden significant decrease in vision. And in more severe cases, those that are associated with fibrovascular growth and retinal detachment, patients can report suddenly losing part of their visual field, such as a black curtain appearing somewhere in their field of view. Actually, all patients with diabetes, whether it's type 1 or type 2, they're all at risk of developing a diabetic retinopathy. In fact, the longer a person has diabetes, the higher their risk. It is estimated that after 20 years of diabetes, nearly all patients with type 1 diabetes and more than 60% of patients with type 2 diabetes will have some degree of diabetic retinopathy. And this is like the most important risk factor for developing a diabetic retinopathy, so the duration of the disease. But there are also other risk factors, among which we can cite poorly controlled or uncontrolled diabetes, blood pressure that is uncontrolled, so hypertension, cholesterol or dyslipidemia, smoking, and pregnancy.
That's really interesting. So I think for me, like, one of the key things I took from that was that, one, people have to go get their eyes checked regularly if they are at risk, so if they have diabetes, but then also controlling all these other sort of your whole health and your whole body factors are also really, really important in trying to manage the disease.
Exactly. It's really a team effort. It's not just the eye doctors that can help patients with diabetes. Like they should follow up with their nephrologist for the kidneys as well, and have a regular checkup with their family doctors for sure.
Yeah, that's a really great message. I'm going to now ask you about your research projects, which I know everybody is probably really excited to hear about. So could you maybe just give us a little overview about your research project? I know it's about microglia, so maybe explain a little bit about that and what you're trying to study.
Yes, of course. So I guess we talked a little bit about the eye already. So the eye is part of the central nervous system, which is composed of the brain, the spinal cord, and the eye. And the central nervous system is in fact formed by neurons on one side and glial cell on the other side. So maybe people are a bit more familiar with neurons, which are the electrical cells of the brain and are responsible for transmitting information in the nervous system. Now, glial cells, on the other hand, were initially thought to only provide structural support to the neurons. In fact, the word glia, which is derived from the Greek, the same word actually, glia, means glue in English, and reflects the impression that these cells were only necessary to hold the neurons together. That's what people used to think at the beginning of the last century. We now know that this is not exactly true. There are several types of glial cells, and they can perform a wide range of functions, from support to protection of neurons, and even formation and remodeling of connection in between neurons. So microglia is one of those subtypes of glial cell, and they're smaller in size than other types of glial cell. And they're tissue-resident macrophages, which means that these are cells that are responsible for detecting, engulfing, and clearing up cellular debris and pathogen. They, in fact, act as primary immune defense of the central nervous system. They're very special because they can adopt a wide range of forms. So in their physiological condition, microglia adopt a ramified form, like a little octopus, if you want, with branches that continuously move and to probe and sense the environment with a variety of receptors on their surface. With those, they can detect the presence of damaged neurons, cellular debris, inflammatory molecules, and other potential pathogens that could be harmful to the central nervous system. And if they do encounter such signals, microglia can become activated and adopt a more rounder form and migrate to the site of damage, where they can release the inflammatory cytokine to counter this tissue damage. Now, in diabetes, microglial cells play an important role in maintaining several immune checkpoints and restoring the balance in between anti- and pro-inflammatory states. However, if conditions where blood sugar is elevated persist for a long period of time, the balance can be shifted towards the secretion of a large proportion of pro-inflammatory molecules, which leads to the dysregulation of microglial cells. And this chronic inflammatory response is then associated with breaches in the blood-retinal barrier, formation of new blood vessels, recruitment of other macrophages, and neuronal cell death throughout the release of cytotoxic substances. So I guess in the recent years, the idea that diabetic retinopathy is purely a neurovascular disease has therefore slowly evolved to suggest that chronic inflammation might also play an important role in triggering diabetic retinopathy. Interestingly, previous postmortem studies of patients with diabetic retinopathy have shown that microglial cells are present in all stages of diabetic retinopathy, and they are often found in their activated, more round form, where they are clustered around the retinal vessels and areas of bleeding and neovascularization. So in this context, we will study the molecular markers and inflammatory molecules that can be released by microglia in different stages of diabetic retinopathy. And by characterizing and comparing the repertoire of molecular biomarkers secreted in the eyes of healthy patients versus the patients that are affected by diabetic retinopathy, we hope that we will be able to better understand the role that microglial dysregulation and chronic inflammation can play in diabetic retinopathy.
That was really interesting. So, like when you were describing diabetic retinopathy originally, you mentioned that when there isn't enough oxygen because the vessels are damaged, the eye makes some more blood vessels. So now, are you suggesting or hypothesizing that not only is this happening, but that also the activation of inflammation in the microglia also leads to increased blood vessel growth? Was that correct?
Yeah, that's right. That's why we're thinking.
Okay, that's really, really interesting. And is there also, I know whenever we talk about inflammation, it's a really popular sort of topic and people get very concerned about, are there ways to like lower inflammation in the body, diet, things like that? Are we talking about the same types of inflammation, or is it a different type of inflammation? So could people reduce inflammation if they have diabetes through, say, through diet or some other exercise or other types of activities?
I guess that's a really important, interesting question. Actually, there's even preliminary studies that have suggested that some herbal medicine, curcumin, which is could target microglia, and there are actually studies looking at that, if curcumin could play a role. So this would be like something that you can take at that is global, but that can have more local effect. But this is really preliminary data. So I don't have the answer for that, unfortunately.
So stay tuned.
Yes.
Yeah. So just finally, before I move on to talk to our next clinician-scientist emerging leader, I was wondering if you could just tell me sort of why you're so interested in studying diabetic retinopathy and why you think the research into this specific disease is interesting and important.
So diabetic retinopathy is the leading cause of vision loss among people of the working age and the fourth leading cause of blindness in the world. In 2020, approximately 10% of Canadians had received a diagnosis of either type 1 or type 2 diabetes. And among these people, almost 25% are affected by diabetic retinopathy, which represents approximately 500,000 Canadians. In the last decade, there has been significant advances in the understanding and treatment of diabetic retinopathy. So therapies have actually shifted from using laser photocoagulation to target those aberrant blood vessels to injection of anti-VEGF agents into the eye, which can help improve diabetic macular edema and stop the formation of these new blood vessels in patients with proliferative diabetic retinopathy. However, it's interesting to note that despite all of these advances, a significant proportion of patients, and this depends on the references, but a number that I've seen quoted is up to 40% will fail to improve or will not fully improve with anti-VEGF therapies. So I think it's therefore important to continue to study to better understand the pathophysiology of this condition. And by gathering detailed information on the molecular biomarkers secreted during diabetic retinopathy, we hope to better understand the role that other targets, such as microglia, might play in diabetic retinopathy, and of course, ultimately develop new therapeutic targets.
Sorry, I put myself on mute. It started to pour rain outside, so it was a bit loud. Yeah, no, it's very clear from the stats you said how important it is that we are always looking for new treatments to help people who maybe can't use the treatments that are currently available. Thank you so much for sharing about your work and a little bit about your career. I'm going to move on to Dr. Tina Felfeli, and then we'll bring you back at the end to answer some questions and answers from the audience afterwards. So,
sounds good.
Yeah. Hi, Dr. Felfeli. How are you doing?
Good. How are you, Dr. Moniz? Nice to see you.
I'm good. Yes, I feel like I'm on an interview panel show. I am on an interview panel show, like bringing people on and off. Yes, I'm going to sort of start with a very similar question I asked Dr. Gobert. If you could tell us a little bit about what brought you to the stage you're at. So I know you're pursuing a PhD at the same time that you're completing your residency. So how's your career progressed to this point?
Yes, thank you so much, and thanks for all the attendees. And so I'm Tina Felfeli. I'm currently a resident in ophthalmology in the Department of Ophthalmology and Vision Sciences at University of Toronto. I became interested in medicine through volunteer activities and working with elderly patient populations. And so starting medical school, I hadn't been very exposed to ophthalmology or vision research at all. I became really interested in research from early on, and I always knew that a clinician-scientist path was very suitable for me. And as I became exposed to ophthalmology and chose ophthalmology as a residency specialty that I wanted to pursue, and was very lucky to get accepted into the residency program to become a resident and an ophthalmologist in the future, I knew at that point that I could really invest the time into research and devote time where I can merge the two worlds of science and medicine and the research that I do on a day-to-day while incorporating that into the clinical aspect of the patients that I see and treat, and taking that information back and incorporating it into my research. So it really seemed like the perfect time. I'm one of the first residents at University of Toronto to pursue a PhD as a part of the integrated physician scientist training program. But I think it has really allowed me to merge the two worlds of research in medicine.
I think that's really great, and I hope that you're an example for other residents who are thinking about doing research to look into doing this combined ophthalmology residency as well as PhD work. So your work is actually quite different than what Dr. Gobert was talking about, and you're looking at, or you're in a public health department, I know. So I was wondering if you could tell us a little bit, first of all, about the disease that you will be studying, which is non-infectious uveitis.
Yes, absolutely. So as you correctly mentioned, so my research really focuses on clin-epi or healthcare health services research. And so I kind of talk about uveitis, which is the disease entity that I'm interested in. And I think this will set the background for a lot of our listeners. And so uveitis is a broad group of inflammatory intraocular inflammatory diseases, and they're quite broad. So there's over 30 of these disease entities that have been identified. And so based on studies to date, its prevalence has been estimated to be anywhere between 38 to 200 per 100,000 people. And so it can happen in all age groups, but primarily it affects people between the ages of 20 to 60 years old. And so there are some studies that actually suggest the annual incidence of uveitis is rising in adults that are between the ages of 20 to 50, with reports ranging between 60 to 80% of all the uveitis cases actually happening in this age group. And so the term uveitis itself, which comes from uvea, which is actually from a Latin word, uva, meaning grape. And so it consists of the uvea itself. It consists of the middle pigmented vascular layer of the eye, and includes the iris, the ciliary body, and the choroid, as you can see in the image, which is depicting the anatomy of the eye. And then the term itis itself refers to inflammation of the uvea. And so together they form the term uveitis. And so this really is referring to inflammation within the uvea of the eye. And it may be involved with other structures as well. So other structures of the eye, such as the retina, the sclera, the cornea, vitreous, and the optic nerve may also be involved in uveitis. We often group uveitis based on the parts of the uvea that are affected. So there's 4 types of uveitis that are typically known. We have anterior uveitis, intermediate uveitis, posterior uveitis, and panuveitis. And these are, again, based on the anatomical locations of the uveitis or the inflammation that's happening within the eye. One of the important aspects of uveitis is that it may be limited to the intraocular structures, or it may be part of a broader systemic disease, such as psoriatic arthritis, inflammatory bowel disease, ankylosing spondylitis, and juvenile idiopathic arthritis. And a lot of these conditions affect patient populations from various age groups. With regards to some of the symptoms, and I think that's important for our audience to know about, they may occur quickly, so what we call acute, and this may last about 6 weeks or so, or in some cases may very slowly occur over time. And in those cases, we refer to them as chronic, and these are ones that last about more than 6 weeks or so. And they can get worse over time quite quickly, and they may affect one of the eyes or both of the eyes or one at a time. And so patients may experience symptoms such as redness, pain, sensitivity to light, blurred vision, floating spots or dark spots, and then general decline in their vision. And then based on the systemic conditions that I mentioned earlier, there may be other systemic manifestations such as joint stiffness, rashes, and oral ulcers that may be present in patients that have uveitis.
That's really interesting. Actually, it sort of goes back a little bit to what Dr. Gobert was talking about the whole sort of body manifestations of some of these diseases, how one, you might have one chronic illness which can potentially lead to the other. I was wondering if you could tell us about how uveitis, or non-infectious uveitis, I guess in particular, is currently treated. And I know, at least for me, this is a relatively new eye disease that we don't talk a lot about at Fighting Blindness Canada. So it'll be interesting, I think, to our audience to understand a bit more about that.
Yeah, for sure. And I think that's a really, really good question. And I'll kind of talk about the fact that first and foremost, I think what's really important to highlight is that uveitis is very serious, and it can actually lead to vision loss if it's undiagnosed, if it's left on treatment. And so it's based on, I think, what was also discussed about the other eye conditions that are being studied in this webinar that we're going to talk about. It's actually also one of the leading causes of preventable blindness in adults in Western countries. And so just to kind of give you a perspective of why it's so important to diagnose and treat patients with uveitis, in some of the developed countries, such as the United States, that are our close neighbors, uveitis causes about 30,000 new cases of blindness each year. And although this information isn't available for Canada, we can get a good sense of how much uveitis is having an impact on patient populations. And so again, it's really important to diagnose and treat uveitis as early as possible, and ideally before there's irreversible damage that has occurred to the eye. And kind of touching on what was already mentioned as well, with the important conditions such as diabetes and diabetic retinopathy that also cause major blindness in patients, uveitis is a lot less prevalent in patients, so about 250 times less common. But comparing it to other conditions such as diabetes and diabetic retinopathy, it actually causes equivalent number of patients to lose vision and lose their sight each year as a result. So one of the main challenges with uveitis is actually the diagnosis and the management of these patients and the complexity because of the heterogeneity of the disease itself. And so we usually, for treatment selection, we start with a very thorough history and overview of symptoms and the presentation, as well as identifying the eye examination features, as well as a physical examination of other organ systems. So this may happen in collaboration with other colleagues, rheumatologists, respirologists, who may be involved in caring for these patients and really localizing the type of inflammation. So these are the first steps before initiating treatment. And then once the anatomical location and the type of inflammation involved is identified based on the eye examination and the physical examination, we may do additional investigation. So those may include laboratory tests. We may do imaging studies. We may do further eye imaging studies. And through all of this, in addition to patients' symptoms, we then come to diagnosis and a conclusion in terms of the best management strategy for these patients. And so I've listed some of the treatment options that we have available for these patient populations, and these are for non-infectious uveitis. And there's a wide variety of medications. There's a wide spectrum of them, and again, for a wide spectrum of diseases. And so the goal of all of these treatments is really to eliminate inflammation, alleviate pain for patients, and prevent further damage to the various tissue organs, including the eye, and then prevent vision loss. And so treatment is really dependent on what the patient is displaying. So in some cases, we may use topical treatments such as corticosteroid drops administered to the eye. Sometimes we may use injections to provide more local treatment within the eye. And then in other cases where this uveitis is quite severe or there's systemic manifestations of uveitis, we may use what we call immunosuppressive agents that may be taken by mouth or they may be administered by infusions. And this is really to take a more broad approach to managing the inflammation. Some of the things that I'll kind of touch upon that I mentioned earlier with regards to the symptoms for the patients, it's really important to recognize that the symptoms will vary between patients. And so some of the signs and symptoms that we talked about, for example, for anterior uveitis, we know that patients have a lot of pain. They may have a lot of redness and blurring. For other types of uveitis, such as intermediate uveitis or posterior uveitis, there may not be any pain, or some of these may be slower in demonstration or presenting themselves, and usually maybe more difficult to diagnose. So usually they require a dilated eye examination by an ophthalmologist to be diagnosed. And so for those reasons, it's really important that patients seek care, are appropriately diagnosed, and then the treatment is started accordingly.
I thought it was really striking when you mentioned the number of people who lose their sight because of uveitis, even though it's potentially less common than some of the other eye diseases we talk about. So in terms of the number of people who are losing their vision, I'm not sure if there's something you can generalize, but is it more because it's so hard to diagnose that by the time it's diagnosed, people have already lost their sight, or is it because there aren't enough treatment options available, or a combination of both?
Yeah, I think it's a combination of both. So these are usually, there's been studies to really show that the indirect cost to the patient, so the burden of the disease for uveitis is very high. A lot of the times, these are very difficult diseases to manage. So depending on the etiology, patients may have relapses or recurrences over time. Their response to treatment that we initiate them on and they respond to well initially may change over time. So they may have to change their treatment options multiple times in order for us to be able to find that optimal treatment for them. And so a combination of all of those various issues really contribute to that vision loss. And a lot of the posterior uveitis disease entities do cause a lot of severe impairment that can be irreversible at times and hard to treat.
Okay, thank you for clarifying that. So I'm going to move on to ask you a couple of questions about your project. Your research project, which is part of your PhD research as well. So it uses three relatively sort of different techniques. I thought we could maybe go through each one by one, because otherwise it will be probably a little bit too much information in one go. So you're using a combination of a technique called economic modeling, artificial intelligence, and something called proteomics to, in my understanding, help come up with a better diagnostic and treatment pathway for patients. So I was wondering if you could maybe start off with proteomics and explain how that part of your project is going to help you manage patients better.
Yes. I really like to kind of divide my research into two different components, and I completely agree. These different methodologies each have their own values and different levels of complexity that I think is really good to go through step by step, as you suggested. And so my research has multiple aspects and methodologies and multiple disciplines, and it, in a sense, combines biomedical and clinical research in ClinEpi. And so on one hand, I like to look at trends in the population. So before we get into the proteomics aspect of it, but just the trends in population eye health. And so in order to answer really important questions, such as what are the number of patients in Ontario that are affected by uveitis? So what is the prevalence of uveitis? How many patients are affected? How are they affected? What are the age groups? Are they being diagnosed in a timely manner? Once they're affected, are they accessing care? Are they able to get the treatments that they need? And so this is really a big picture overview of the disease and its burden, and really provides context for what else can we do and where can we implement strategies to better care for these patients. And it uses data for the entire Ontario population. And so then looking on the other hand, the other aspects of my research, kind of diving away going away from this kind of bigger picture aspect to a more personalized approach to medicine, I like to look at patients at the individual level. And so if we look at the specific individual within the population of Ontario, what are the specific qualities or characteristics about this person that make them unique? What is it about them that makes them non-responsive to a treatment option that other people may respond to? And how can we use various characteristics or unique qualities about them to better optimize or personalize their treatment? And so that's another aspect of my study, and that's where really proteomics comes into play. Now, I'll talk about proteomics in a little bit, but because both aspects of a population-based approach and looking at proteomics and individual level data, there's a lot of data that you look at, but even within the individual level, there's various characteristics of the patient population, inflammatory markers, and then within the population, you're obviously looking at many individuals as well. And so what really becomes helpful in both those scenarios is the handling of the data. And in this case, artificial intelligence or machine learning, which is a buzzword that many will have heard nowadays more recently, are very helpful in looking at meaningful trends in all of this data, and how do we sift these, put these data together and make meaningful trends and meaningful ideas that can be derived from some of this information. So for example, the proteomics that we talked about, which is looking at the inflammatory biomarkers in a very close-up look at what they do in uveitis. How do they play a role in the disease progression, and also how are they lowered when we treat patients with various anti-inflammatory medications, and how they may serve as new targets for other treatment options that we may introduce. This is where machine learning becomes really useful, and it combines the patient data, their outcomes data, their biomarkers data all together, and provides suggestions for personalized treatment options for patients. Now, the other part of it that you also mentioned was economic evaluations. And so economic evaluation is really important, I think, in the Canadian healthcare system context. So we spend a lot of money in our universal healthcare system for treating and managing various diseases. And uveitis is one of them. It's one of the really important conditions that we also spend a lot of healthcare resources on. And so what's really important and I find interesting is identifying how to best allocate our healthcare resources and spend our healthcare dollars on what is really most important. And so if we do that, if we shift our focus into spending our healthcare dollars the best way, we can actually invest in new treatment options, new management strategies, rehabilitation for patients, so that healthcare dollars or healthcare resources can be better invested elsewhere once we identify the most cost-effective treatment options. And so for this part of my research, I use another methodology, which is called microsimulation modeling. And you may actually heard about microsimulation models through your TV or your news for resource allocation in terms of COVID vaccines or numbers for COVID. And we use modeling a lot for prediction reasons. And so in the same way, microsimulation modeling for assessment of treatment options for uveitis is really useful in terms of using various parameters and a very complex mathematical model, and give us predictions of how effective these treatments are. Are they cost effective? How can they best be used? And it gives us a guideline in terms of what other ways we can best allocate our healthcare resources. And so I think overall, to kind of sum up everything that I've talked about, really what this research aims to do is use different methodologies. So we talked about population level details, we looked at specific individual level details, and then looking at machine learning and economic evaluations to be able to best treat patients, diagnose them, and ultimately prevent blindness in our patient population.
Thank you, Dr. Felfeli. That was a really nice overview of a pretty complex project. So I really appreciate you breaking it down for the population level, the individual, and then how you bring all that together to work within our healthcare system. That was very impressive, and I'm looking forward to hearing more about it in the next few years. So I'm going to move on now to our final clinician scientist, Dr. Stephan Ong-Tone. How are you doing? You sort of joined us a little bit later, so I'm glad you were able to join us.
Yeah, thank you very much for having me. Sorry, I was just finishing up clinic there, but
of course, doing really important things. That's more than okay. I'm glad you were able to make time to join our webinar today. So yeah, the same as with the other awardees. It'd be great if you could just tell me or tell us a little bit about your sort of career path and how you went from going through med school, doing some research, and ending up where you are now, which is as both an assistant professor as well as a clinician at Sunnybrook.
Sure. Well, first of all, I'd like to thank Fighting Blindness Canada for this opportunity and recognition for this very, honor to be awarded this award. And also congratulations to my other co-recipients, Dr. Felfeli and Dr. Gobert, who I know both personally, and it sounds like they're doing some very exciting research. So I'm really looking forward to what they're going to be studying. So a little bit about myself. So my name is Dr. Stephan Ong-Tone. I'm currently a cornea specialist and clinician scientist here at Sunnybrook Health Science Center and Sunnybrook Research Institute. A little bit about my background. So I did my med school, my MD, PhD at McGill University. I actually did my PhD in neurological sciences at the Montreal Neurological Institute. And at the time, I was very interested in CNS nerve regeneration and look at regenerative strategies to promote functional recovery after spinal cord injury. And I was actually kind of destined to kind of go into a career in maybe neurosurgery, but then I got exposed to ophthalmology and really saw, how, important people's vision can be and how much of an impact you can have in someone's quality of life by addressing their vision. And also I saw that there was a lot of amazing opportunities for research and for basic science research and for translational research and potentially to apply some of the concepts that I've studied during my PhD, for ophthalmology. So I came to University of Toronto to complete my ophthalmology residency, which I completed in 2018. And after writing the board exam, I decided to pursue a cornea fellowship. So I went down to Massachusetts near Harvard Medical School for a 2-year clinical research fellowship. Where I really got to dive back into the research, where during the first year of the fellowship, I really got to focus on basic science research on corneal endothelial cell therapy and investing Fuchs dystrophy, which I'll talk about a little bit more, but really laid some of the scientific groundwork, I think, for some of the studies that I'm proposing in this proposal, but also in terms of establishing my lab and my research program here at Sunnybrook Research Institute. And I also did my training there to be a cornea specialist and to be able to do corneal surgery. And I think it was really at that time where I was really convinced myself in terms of my decision to go into a career as a clinician scientist, because I found myself, and maybe I don't know what the other recipients thought, but throughout my training, I've always been interested in both in scientific research and clinical work, and I've been back and forth a lot, in terms of how much I'm going to pursue each. And I think I'll give you a little anecdote during my fellowship. It was really one day where in the morning, I was doing corneal transplants on patients with Fuchs dystrophy, myself doing the surgery. And then I was actually able to take the specimens from these patients and take it the same day to the research lab that I was working in, and to do the experiments and to to do the experiments on these cells. So I thought that was pretty cool. And I remember reflecting, on that day of the unique opportunity and ability, to be, at the forefront of both clinical research, basic science research, but also providing patient care as well. So it's a very humbling opportunity. I just started here at Sunnybrook back in September, October, and things are underway. It's crazy to think it's almost been a year, but the research program is kind of up and running, and we're seeing patients. So I'm excited to be here, and thank you again for this opportunity.
Oh, thank you for that that description. That was wonderful, and it must be a really exciting point in your career to be starting your own lab as well as getting your clinical practice underway. You mentioned a few times Fuchs, and so I know you're working on a disease called Fuchs Endothelial Corneal Dystrophy. I was wondering if you could tell us a little bit about that specific disease.
Sure. So I think before talking about the disease, I have to take a step back and kind of explain a little bit what the cornea is. So the cornea is kind of the front part of the eye that allows us to see. And the cornea has 5 different layers to it. And if you start kind of from the most superficial layer, there's the epithelium, there's a specialized basement membrane called Bowman's layer, there's a stroma, and then there's the inside of the cornea, which is the corneal endothelium, which lies on a specialized basement membrane called Descemet's membrane. And that primary function of that inner lining is basically to keep the cornea clear. So if you think of the eye as kind of filled with fluid and it's filled with aqueous, and there's a certain pressure within the eye, if that inner lining wasn't there, basically what would happen is the cornea will swell up and become filled with fluid. So the primary function of this corneal endothelium, which are composed of endothelial cells, is to act like a pump. And these cells, what they do is they pump fluid out of the cornea to maintain corneal clarity. And basically, when these cells become dysfunctional or start to die off, that the cornea basically loses that ability to remove fluid from the stroma, or the middle of the cornea, and the cornea becomes basically filled with fluid, and it can result in a reduction in vision and eye pain, basically. So Fuchs endothelial corneal dystrophy is actually the most common corneal dystrophy, endothelial corneal dystrophy, and it's actually the leading indication worldwide for corneal transplantation. It affects about 4% of individuals over the age of 40. Okay, and we know it's an age-related disease that tends to affect patients, anywhere starting in their 50s to 60s to 70s. And we know that there's also a female predilection of it. About 3/4 of the patients that we end up seeing are females. And we know there's many different causes that lead to Fuchs that can contribute to it. We know that there's environmental risk factors such as UV light. We know that there's also a strong genetic, basically, predisposition to this condition. If we look at the genetics of Fuchs, we know that in some cases, up to 79% of patients have this genetic mutation. It's in this gene called TCF4, transcription factor 4. That they have this sort of genetic mutation that predisposes them to this disease. And it's actually interesting that if you look back on, so Fuchs, what's this disease named after? It was first described kind of in the early 1900s by an Austrian ophthalmologist called Ernest Fuchs. And at that time, technology was limited. So basically, all he could see was that the cornea was swollen. And it was actually first described as more an epithelial disease because we couldn't really look inside the different layers of the eye. And it was actually thought to be more of a corneal swelling disease. And then the epithelium was more affected. But then, as technology improved and our ability to look at the different layers of the cornea, we were actually able to, ophthalmologists were actually able to further describe and see that the dysfunction was actually on this inner lining of the cornea, the corneal endothelial cells. So that's what Fuchs is. It's basically this disease that causes damage to these corneal endothelial cells that start to die off, and patients present initially with could be very mild symptoms. Actually, very early on, it could just be diagnosed by an eye care provider, and the patient could be quite asymptomatic. And over time, patients will present with reduction in vision, glare, and decreased vision, and eventually, if the cornea becomes so swollen, it eventually leads to eye pain.
So, are there any treatments currently for Fuchs?
Right. So currently now, in terms of the treatments, I like to think of things as kind of treatments as sort of medical management and surgical management in Fuchs. So in terms of medical treatment, it's not really a treatment. It's more of symptom management. So as I was describing in Fuchs, basically the cornea becomes very swollen, and what we use as a treatment for this, or kind of to manage this corneal swelling or corneal edema, is a hypertonic saline drop. So basically, high salt solution that's made into sort of an eye drop that the patient basically administers to their eye. And the idea is that by putting a high salt solution, it kind of pulls out some of the water from the cornea to try to reduce some of that swelling. But there's only so much that can do, and eventually there's so much cell death, and there's not enough cells there to kind of pump fluid out, and there's only so much the hypertonic saline can do, that eventually now the next step is usually a surgical intervention. And kind of the standard of care now is endothelial keratoplasty, or corneal transplant. And basically what that means is that we remove the abnormal cells. So just like a partial thickness corneal transplant, we remove the abnormal inner lining of the cornea with its basement membrane and replace it from a donor with basically that same anatomical replacement of just normal corneal endothelial cells with their basement membrane, which is called Descemet's membrane. And that's sort of the standard of care now. There is a sort of newer treatment that's being investigated for early Fuchs, and it's only certain type of patients that qualify for this new procedure. Is basically removing the abnormal corneal endothelium only if it's restricted to the center of the cornea. And we can actually let the quote unquote normal cells migrate from the periphery to regenerate the corneal endothelium. And then this obviates the need for a corneal transplant. And there's certain, because there's certain risks associated with any sort of surgical intervention.
I actually want to go back 1 second just to ask a couple of follow-up questions about what you were talking about how it's more common in females. I was just wondering if you know why that is.
Right. So this is so there's some evidence actually in the lab that I last worked in Boston. One of the theories, and we showed this in sort of an animal model of Fuchs, is that we think that estrogen, higher levels of estrogen, which is obviously higher in females, leads to some sort of toxic pathway that leads to some DNA damage. That causes more oxidative stress and mitochondrial dysfunction to eventually lead to more cell death in these corneal endothelial cells. But it's been shown in sort of animal models, and we showed it in sort of some human patient specimens, but that's sort of the current hypothesis and current evidence suggesting why there's a female predilection of Fuchs.
Interesting. And you also mentioned that there is one genetic sort of predisposition, TCF4, I believe it was.
Yes.
So when you say that is a predisposition, does it mean, so if you have that gene mutation or a mutation in that gene, are you definitely going to get Fuchs, or it just increases the risk that you will get Fuchs?
Yeah, so it increases the risk that you have it. There have been studies, there's a lot of these population-based studies looking at the genetics of Fuchs. Obviously, you look at normal controls, and in about 4% of normals, patients without Fuchs disease, they do have this genetic mutation. So the question is always whether, if you follow these 4% of patients, down the line, are they eventually going to develop Fuchs? We don't really know that 100% for sure, but we know that if you look at patients with Fuchs disease, and if you look at the genetics, we know that a large percentage of them have this sort of genetic mutation in it. So that's what really is kind of predisposing them. And that's been the strongest genetic mutation that's been linked to Fuchs disease, especially the age-related onset of Fuchs disease. There are, age-related where Fuchs that tends to affect patients in more their 50s and 60s and 70s. There's other rarer forms of Fuchs disease, kind of an early onset, where there's known mutations in collagen where patients present a lot earlier, but they seem to be kind of different, slightly different presentations. But the more common one is this age-related Fuchs disease with this genetic mutation.
Thank you. Yes, now let's get to, I'm sure everybody wants to hear about, which is your research. So your project is to test out a gene therapy approach as a potential new treatment for Fuchs. I was wondering if you could tell us just a little bit about what your project is about and how you're going to use a gene therapy approach.
Right, sure. Well, I'll talk a little bit about gene therapy kind of in general and what that is, just kind of a synopsis. So basically gene therapy is developing some sort of approach to basically target some sort of genetic defect. So in certain cases, the gene is deficient, right? So just in general, where the body has some sort of genetic mutation where it doesn't produce the gene of interest, doesn't produce the protein of interest. So in that case, gene therapy would basically mean that you're trying to replace the gene that's missing. So that's one form of gene therapy. Another form of gene therapy, and this is kind of where Fuchs kind of falls in, is kind of like the genetic mutation is present in only the affected individuals. So the strategy is basically to try to remove this genetic mutation to make it more like a normal gene. So my project is kind of looking at how does this genetic mutation kind of contribute to Fuchs pathogenesis? Because we know that, like I mentioned before, about 79% of patients, or up to 79% of patients with Fuchs can have this genetic mutation. And there's some evidence in terms of how this contributes, but it hasn't really been well elucidated. So part of my project is trying to understand how this genetic mutation contributes to Fuchs pathogenesis. And the way that I'm approaching this is actually using these reagents that I developed at Harvard. So one of the challenges of using patient specimens to doing research directly on it is that basically when you do surgery and you collect specimens and you try to do an experiment, you don't have that many cells, and you're kind of limited in terms of the number of experiments you can do because you basically only have that one tissue to do kind of one experiment. So one of the ways that we kind of have developed, and some of the tools we've developed to address, this methodology, and it allows us really useful tools to ask very good scientific questions, is so what I've done is that I've taken specimens that have from Fuchs patients that have undergone corneal transplants, and I've taken them to my lab, and we actually immortalize them using something called an SV40 immortalization. But basically now we get these cell lines that we can do many, many experiments from to ask different scientific questions. And at the same time, we can genetically, we can genotype, so look at the genes and kind of classify these different cell lines based on their genotype. And so now I have a kind of a library of these different cells that were taken from different individuals with Fuchs that have different genetic mutations, or we know the genetic status. So now I can do multiple experiments looking at different readouts to look at how these different genetic mutations, or maybe their sex, how does that affect, potentially can affect Fuchs pathogenesis. So that's kind of the first part of my project, is trying to investigate how this genetic mutation contributes to the disease process. And the second part of my project is trying to develop a therapeutic, thinking as a translational approach, is how can we actually maybe target and remove this genetic mutation that, in theory, can contribute to Fuchs pathogenesis. Can we develop a gene therapy strategy to remove this genetic mutation so that we can offer it as a therapeutic to patients going forward? And how I kind of envision this is that maybe going forward, we can identify early on Fuchs patients without, very severe disease manifestations. But we can know that they have a genetic predisposition to this disease. So maybe we can target them early on, genetically modify some of their corneal endothelial cells and transplant back in those genetically modified cells as a sort of personalized medicine for that specific individual, that they may not develop Fuchs going forward.
That's really, really interesting. So has anything like this ever been tried before? So using a gene therapy approach to try to cure a corneal disease?
Nothing that I know of in terms of a clinical trial and in terms of that's current like standard of care for a corneal disease. There definitely have been studies looking at different animal models, looking at gene therapy using the same sort of strategy of using adeno-associated virus, but nothing is really for corneal disease. Mm-hmm. There is a currently it's been approved for Health Canada approved for inherited retinal diseases using this adeno-associated virus for RP and I think Leber's congenital amaurosis. And actually, Sunnybrook here is one of the 2 Canadian sites that are approved to deliver this this therapeutic. So it's kind of exciting place to be and to hopefully develop this therapeutic in my lab and hopefully translate this to a clinical trial so that we can help patients with Fuchs dystrophy.
Definitely. The therapeutic you're talking about is called Luxturna. I know a lot of our community will know about it because we were so excited when it was approved by Health Canada, and we're now just waiting for patients to get access to it. Some of them hopefully at Sunnybrook soon. Mm-hmm. Thank you so much, Dr. Ong-Tone. I'm going to sort of turn it back to Morgan, and maybe all of the awardees can come back on to the screen and get some audience questions.
Awesome. Well, thank you so much, Larissa, and thank you to all of our scientists, Dr. Gobert and Dr. Felfeli and Dr. Ong-Tone. This is really what Fighting Blindness Canada is all about: is learning about this new research, funding this research, and hearing about it is just so exciting. So we do have an opportunity now. If anyone has any questions, you can put them in the Q&A box at the bottom of your Zoom window, or you can email me at education@fightingblindness.ca. So I have a few questions that have already come in over email. So let's go through those. So I think the first one here is for Dr. Ong-Tone. I will apologize. This question came in Italian, so I'm using a Google Translate. So hopefully it makes sense to you. This is from someone named Montefiore. He says, hello, my daughter is affected by Fuchs syndrome in her right eye, diagnosed as non-chromatic monocular, and manifests itself with endothelial precipitates that disappear spontaneously or with anti-inflammatory or cortisone drops. The syndrome should be viral in origin due to the daughter's prematurity, but his question is whether or not it might be able to be treated with gene therapy if a treatment was to become available.
Well, that's interesting. I mean, obviously, in terms of the diagnosis, typically these guttae, these collagenous deposits, when they do form, they don't typically reverse, and they're not usually steroid responsive. So the fact that they are steroid responsive, sometimes there's other mimickers of Fuchs dystrophy that present with what we call pseudo guttae. So that I'm not sure, So with respect to the question of whether this can be treated with gene therapy, I mean, it would be kind of depending on what the underlying condition is and the sort of genetic mutation is, so, and currently to sort of develop some sort of therapy is actually a very long pipeline, to develop that. But I think in general, I think gene therapy is something that's very topical now, and there's a lot of research that's going into it, and not only gene therapy within the eye, but just gene therapy in general. That I think there's a lot of promise for the future. That if there is a specific genetic mutation that can be reversed, then I think in theory, then yes, it could be targeted by gene therapy.
So would you recommend that people who are affected by Fuchs have genetic testing to try to get to the root of what their mutation is?
Yeah, so I mean, that's a very interesting question. I mean, as a researcher, I think that's yes. I mean, I think it would be great if we can test everyone and we know that genetic mutation is. I think one of the challenges now is that, and this is something that's really interesting, I can, thinking about exploring, is that, you know, there's no easy avenue in terms of how to do genetic testing for Fuchs. It's not like, you know, there's a place where we can just send off blood and do genetic and look for that mutation specifically for Fuchs. So I think it would be very valuable, and that's something that I'm doing more of on a research basis of, you know, the patients that I'm seeing with Fuchs dystrophy, of collecting their blood and doing some genetic, you know, I will be doing some genetic, you know, testing and trying to identify the mutation, because there are many different mutations that have been described. I was really talking about the main one, but there's some other less common ones. But I think going forward, you know, I think it would be very exciting if that was like a standard and pipelined also as well, that anybody can basically send off their blood or a sample for genetic testing specifically for different Fuchs mutations. The question is also, you know, who's going to fund it also as well, and as a lot of these, you know, these treatments and diagnostic tests, you know, come into market, you know, what OHIP is going to cover and what costs are going to be kind of relayed to the patient, and what sort of funding structure there's going to be to kind of support these, you know, important diagnostic tests.
Yeah, it's so interesting because, you know, it's always shocking to me how many mutations can be the cause of the same disease. So, you know, with RP, I think there's like over 50 mutations or whatever that have been associated with RP. So as the research progresses, there's always sort of new things coming out. And, you know, we talk to people who've had genetic testing maybe 15, 20 years ago. Well, it's time to maybe get it again because things have changed over time. So hopefully your research will help to illuminate some of that. While I still have you, I have another question here from Joan. And her question is pretty specific. She's saying, where in Ontario near Guelph could I find a doctor that treats Fuchs? But maybe just in general, if someone has this diagnosis, how can they best find a doctor that could help treat them?
Right. So, I mean, it kind of depends in terms of, you know, the staging of Fuchs. Usually early on, any ophthalmologist could kind of make the diagnosis and start currently, you know, medical management of starting these hypertonic saline drops, and start following you along. But Fuchs dystrophy is typically managed, especially more in the more advanced cases, by cornea specialists that do corneal transplantations. So most, I don't know someone specifically in Guelph, but most larger cities have, you know, ophthalmologists and specifically cornea specialists to help see these sort of patients. Yeah, and I'm obviously happy to see anybody if they want to come over to Sunnybrook.
That's great. Oh, and we just have one more question that's come in for you here through the chat. So Peter says, Dr. Ong-Tone, how many different types of mutations have been identified that may underlie Fuchs dystrophy? Is there more interest in the mutated transcription factor that will be more promising than the other mutations you have identified in your patients?
So that's a good question. I don't know the exact number off the top of my head, but there's probably at least, you know, 15 or so different mutations that have been described. Some of them are different mutations within the TCF4 gene, and some of them are actually different genes that have been identified as well. You know, so the majority, the majority of these cases of Fuchs, when we do the genotyping, is in this TCF4 repeat expansion. But there are other forms of like, and I was alluding this to as well, of early onset Fuchs, where there's mutations in the collagen gene that manifests itself. So in those cases as well, that might be amenable to gene therapy as well. But it's the minority of patients that have this condition. You know, the majority is this repeat expansion in TCF4.
Great. And last question, and then I'll move on. But is there any way that Fuchs can be prevented? Are there any preventative measures that people can take?
Yeah, that's a great question. So there hasn't been anything that's really been definitively shown, and that's currently an area of active research, and I think would be very helpful and promising to a lot of patients. But we know that there is a correlation with UV light and exposure. So in general, I counsel my patients to wear sunglasses when they're outside, UV protection, to kind of minimize the UV exposure. There's other things that have been linked to it as well, such as oxidative stress. So having like a healthy diet overall, eating a lot of green leafy vegetables, not smoking, all those things have been proposed kind of on a population level to have an impact in Fuchs. But unfortunately, there's no like anti-Fuchs drop that's been developed that can kind of prevent the disease. Which is, I think, an area of active research that a few groups are exploring.
Wonderful. Thank you so much for all of those answers to these questions. I'm going to move on to Dr. Gobert. And I have a couple questions here. This one's a little bit more general about diabetes. So if I have diabetes, how often should I have an eye exam? Are there particular tests that are more important for the diagnosis and management of diabetic eye diseases?
So I guess it depends on what type of diabetes, because there are screening guidelines that are slightly different depending on whether you have type 1 or type 2 diabetes. But as a general rule, a regular annual exam would be enough, unless there are complications and more advanced stage of the disease that require more frequent exams. And the second part of the question, I believe, was, are there any specific tests that are required? So I guess the most important is to have a complete eye exam with dilated eye exam that could allow proper visualization of the retina. Oftentimes, we would also recommend the patient to undergo imaging of the retina just to make sure that there is no fluid under the macula, which is the central part of the retina that I talked about a little bit.
Thank you. And someone else has asked here, what is the difference between diabetic retinopathy and diabetic macular edema? And do you think that microglia play a role in both of these conditions?
That's a very interesting question. So I guess diabetic retinopathy is the general term that engulfs all the forms of diabetic retinopathy, either the non-proliferative form, the proliferative form. Now, diabetic macular edema can happen at any stages of those diseases. Actually, the classification is a bit more complex. I didn't talk about all the different stages. Like, for example, in the non-proliferative form, there is the mild, moderate, and severe forms. But it's the most important part is that diabetic macular edema can happen at any point in those phases of diabetic retinopathy. And so, yes, it's possible that microglia could play a role because, like I said, a microglial dysregulation can perpetuate a cycle of chronic inflammation in which there is a break in the blood-retina barrier, which then will allow fluid to accumulate in the retina and cause edema.
And another question here, what can I do if I have diabetic retinopathy and it's progressing to till vision loss? So once that vision loss starts happening, what are the best things that people can do?
I'm not sure if the question relates to the diabetes in general or the diabetic retinopathy, but like the diabetic retinopathy. So as we discussed, like one of the main treatment now for diabetic retinopathy, if there is neovascularization, if there is diabetic macular edema, is injection into the eye, so intravitreal injection of anti-VEGF agent, which will help in regressing the new blood vessel, the aberrant neovessels, as well as the macular edema. So I don't know if that answers the question.
Yes, absolutely. And is that something that someone should see an optometrist for, or an ophthalmologist, a retina specialist? Who should people be seeing when they have diabetic retinopathy?
As of now, in Quebec, so optometrists can diagnose diabetic retinopathy, but they are not allowed to do injection, intravitreal injection. I am not sure. I think there was some debate about whether optometrists should be allowed or not to do those injections, but as of now, I don't believe that it's allowed in any other Canadian provinces. So for that, the optometrists would usually refer to the ophthalmologist, and other people could correct me if I'm wrong, but I think it's also the case in Ontario.
Yes, sorry, I just muted myself. Yes, I believe it is the same here as well. So last question, Dr. Gobert, do microglia play a role in other inflammatory eye diseases or other diseases in general with inflammation, of the inflammatory component?
That's a very interesting question. Actually, there is also evidence now that microglial reactivity is a common hallmark of several other retinal degenerative and inflammatory diseases, and that include age-related macular degeneration, uveitis, and even glaucoma. So this is why investigating the role of microglia in those diseases could have like broader therapeutic implication in the long run.
I think there's one more question that's just come in here. Let's see. Is there any specific pro-inflammatory cytokine that is a more attractive target to treat diabetic retinopathy by downregulating its expression?
That's a great question. Actually, so there were already like other similar studies that were done in Germany, and they have shown that there are actually several cytokines that are upregulated in diabetic retinopathy that include IL-6, so interleukin-6, 8, TNF-alpha, and IL interleukin-1 beta. As of now, I don't know if there would be one that would be more attractive as a target. I think an interesting therapeutic approach would be something that would target a microglia rather than just the expression of those specific cytokines to have a more broader therapeutic effect. Which is why I mentioned briefly that some drugs like the curcumin could have an effect on the microglia, but this is all like in vitro work. There is no evidence for that in vivo, as far as I know.
Well, thank you so much, Dr. Gobert, for answering all of these audience questions. Okay, Dr. Felfeli, I'm coming to you now. So I have a few. So there's a question in the Q&A, and they've said, "Is iritis?" I think they mean uveitis. Let's just see. Is uveitis associated with central retinal vein occlusion?
Yeah, thank you for that question for the audience. It's actually, that's a very interesting question because typically when we kind of think about retinal vein occlusions, we kind of think about the systemic risk factors. So usually things like diabetes, hypertension, dyslipidemia, which cause retinal vein occlusions. But there's actually been some evidence to suggest that chronic inflammation, whether local or systemic, may actually have a role in, have been proposed to have a role in retinal vein occlusions as well. And so that may be due to causing more hypercoagulation systemically due to the chronic condition. So many of patients who have iritis may have chronic iritis, they may have recurrences and relapses. So that potential of association from a systemic inflammation chronically and potentially locally could be there.
Thank you. Are there other diseases that are associated with uveitis?
Yes, absolutely. There's many actually. So we talked about kind of uveitis being isolated to the eye, and there's a number of diagnoses that are isolated to the eye. So, we have general terms for them like birdshot, multifocal court retinitis, AMP, etc. And so those are the ones that are isolated to the eye or maybe associated with systemic conditions. And some of them I mentioned earlier. So Behçet's, Crohn's, psoriasis, ink spot, rheumatoid arthritis, JIA in a lot of younger patient populations. And so their VKH, these are all kind of diagnoses that are associated with uveitis. And so a lot of the times, actually, for these patients, there are systemic manifestations. So if a patient, for example, has sarcoid, their systemic manifestations of sarcoid may be well managed while they're on their systemic therapy, their biologics that they're on. But the challenging part of it is really treating the uveitis. So the manifestation of the inflammation in their eye is actually even more difficult to treat. And so ophthalmologists really play a major role in watching the progression of the disease in a lot of these patients by doing an eye examination and then targeting the treatment and optimizing the treatment that they're on based on the eye manifestation. So absolutely, there's multiple diagnoses systemically that are also associated with uveitis and hard to treat.
It's really interesting. It's a condition that we don't talk a lot about, so I'm learning so much today. Another question here that we have is that, you mentioned non-infectious uveitis, and that's what your project is about. Is there something called infectious uveitis, and does it affect patients in a similar way?
Yes. Very, very good question. So actually, the two main classifications of uveitis that I didn't get into, but it's infectious and non-infectious. So of course, for whatever we have none, there is another version of it. So infectious uveitis is actually quite different in the sense that the treatment options that we use are different. So for non-infectious uveitis, we really try to decrease the inflammation. And patients with infectious uveitis, we may also have to decrease the inflammation and target the inflammation because sometimes due to the infection, it becomes really severe as well. But really the goal is to treat the infectious cause, so whether that's a viral, bacterial, etc. And so you may think of things like syphilis, HIV, CMV. Those are kind of the diagnoses that we're dealing with where we're looking at infectious etiologies of uveitis. And the patient population, the target of the populations that you're dealing with are a little bit different. So we kind of talked about, the patient population that ranges from very young to older. And, when you think about infectious diseases, you may kind of think about patients that may be at higher risk or immunocompromised, et cetera. So it's a different patient target as well. And then in terms of chronicity and acuteness, a lot of them may be more acute than chronic, but again, chronic cases, if there's this long-term infection that they're dealing with, may be present as well.
So if someone is exhibiting symptoms of uveitis, what should they do? Who should they see?
Yeah, and that's the challenging part because uveitis is very tough to know from the patient's perspective. So a lot of the times it may be a very chronic, slow progressing change over time. So they become increasingly sensitive to light. They may feel like they're not seeing as well, especially things like posterior uveitis happen slowly over time. Patients who have anterior uveitis may experience a lot of eye pain and redness. And so really what you want to keep in mind, if you're someone who has a systemic condition that has been associated with uveitis, you should be having an eye examination by an ophthalmologist or optometrist just to ensure there's no disease manifestation intraocularly. And then those who don't have a known diagnoses, as soon as you develop any of your symptoms, you want to seek access to a eye care provider, whether that's your optometrist or ophthalmologist. And then from there on, you may be referred to additional specialists, especially if this is your first time presenting. So you may see a respirologist, you may see a rheumatologist, an internal medicine specialist to really hone in on that diagnosis and what's going on.
Great. And last question, I think, because we're almost out of time. So if someone has uveitis, has been diagnosed with uveitis, are there any lifestyle or dietary changes that the patients can make to help reduce the symptoms?
Yeah. So I think from the eye perspective, really, and the uveitis in general, uveitis is, affects the entire body system depending on the diagnosis that you have. And so really managing the disease itself is critical. And a lot of the patients with uveitis feel a lot of pain and discomfort depending on how their uveitis or their systemic disease is affecting them. So patients with psoriatic arthritis or RA or ink spot may experience joint pain, stiffness, And so really rehabilitation and activity really helps actually decrease a lot of inflammation and help with mobility and preventing the long-term complications of stiffness and inability to mobilize systemically resolve and improve over time. So really managing the disease itself through medications is the best approach, and then implementing some additional strategies such as rehabilitation and exercise in order to manage your systemic disease.
Great, thank you so much to all of our clinician scientists, emerging leaders, Dr. Felfeli, Dr. Gobert, and Dr. Ong-Tone. It's been really, really interesting today, and thank you for just being generous with your time. It's been wonderful to hear about all these great projects, and I look forward to hearing more from all of you over the coming years.